Hello, everyone. I hope that you are all doing well today. Today, I will be presenting an SOP or standard operating procedure on how to use pipettes. So, what is a pipette? I know that a lot of you may already be familiar with pipettes and how to use them, but I think that today would be a good opportunity for us to discuss and all get on the same page about how to properly use pipettes in a laboratory setting. And for those of you who do not know, a pipette is a small tube that is used to withdraw and transfer fluids from one vessel to another. And by fluids, I mean liquids and gases. And the word pipette originated in France and it means tube, which kind of makes sense since pipette kind of makes you think of pipe or tube. And pipettes come in all different shapes and sizes to be able to transfer different volume ranges of liquids and gases and be able to suit different needs in the laboratory setting. And there are four main types of pipettes. There are disposable or transfer pipettes, there are graduated or serological pipettes, there are single channel pipettes, and there are multi-channel pipettes. And this picture here is of a single channel pipette, and it is an Eppendorf pipette, which is a very fancy and also expensive pipette that you may often see in the lab. And this link here explains how to use different pipettes. So I will go ahead and show you this link and show you what the different types of pipettes are and pictures of them and how to use some of them. So first, there are the disposable or transfer pipettes, which are the most basic type of pipettes, and many of us may have encountered them or used them at some point, like in K-12 lab classes. And these are very simple to use. All you have to do is first squeeze the bulb at the top to let out any gases that are already inside of the pipette. Then you put it inside of a liquid or gas solution and then draw out the liquid or gas from that container by slowly releasing the bulb. And then to transfer into another container, all you have to do is squeeze the bulb to eject the liquid or gas. And a graduated or serological pipette is basically a fancier version of the disposable or transfer pipette. And the reason it's called a graduated pipette is because these lines, which measure volume of the liquid, are very similar to the lines, well, they're the same as the lines that you would see on a graduated cylinder. And then, of course, there are the single channel and multi-channel pipettes, which are very similar. The multi-channel pipette is basically a single channel pipette, but with multiple pipette tips. And it's basically the purpose of multi-channel pipettes is to make it easier to transfer multiple volumes at once, especially like, for example, on these 96 well plates. And I will be going over mainly how to use single channel pipettes, just because that's the type that we're going to encounter and work with most often in the lab. So now I'll talk a little bit about the single channel pipette calibration process. So it's a bit of a complicated process, which is why pipettes are typically calibrated by professionals and not by us. And manufacturers recommend pipette preventative maintenance and calibration services at least once every 12 months. And then this link here is for what is a link that explains single channel pipette calibration in more detail if you're interested. And of course, it's important to make sure that the pipettes are clean, both in the interior and on the exterior. And the manufacturer typically will come in at least once every 12 months to do a liquid leak test and inspect the pipettes and be able to recommend different types of pipette preventative maintenance and replace common preventative maintenance parts such as the seal, and also lubricate the piston assembly, and of course perform calibration adjustments as needed, which is a last resort after all of the components are deemed good. And one thing that I recommend that we have in the lab for the pipettes is a pipette calibration log. Basically, you record the serial number of each of the pipettes, the calibration specialist who came in to calibrate them and look at and inspect the pipettes for any sort of damage or issues, and the date of the calibration, and whether or not there was a pass or fail involved with the calibration process, and calibration comments. So I think this might be good to have at least at one place in the lab space. So these are very important notes to keep in mind with the single channel pipettes, and I'll explain some of these notes in more detail during the single channel pipette demonstration that I'll give later in this video. The number one important note to keep in mind is to make sure that the pipette tips match the intended pipettes based on the volumes that are meant for the specific pipette tips, and also the volumes meant for the specific pipette itself. So when you look at a pipette, on the single channel pipettes, it'll show you what volume range it is meant for. And this can range from 0.1 microliters, which is very, very small, to 2.5 microliters, which is also very small. And then other ranges include 2 to 20, 20 to 200, 100 to 1,000, and 1,000 to 10,000 microliters. So for example, one thing that you do not want to do, if, for example, you're trying to pipette 100 microliters, you do not want to use pipette tips that are only intended to transfer 2 to 20 microliters, because if you exceed 20 microliters, then the rest of the liquid will end up inside of the pipette, which we do not want. And that means that we'd have to bring in a calibration specialist right away. And of course, it can be very pricey, not only to buy the pipettes, but also to maintain them. And then another very important note to keep in mind is to go nice and slow. So this picture here from this previous slide is a picture of a very nice fancy Eppendorf pipette. And at the top, you'll notice that there's a little plunger. So what you do is you use that plunger to eject and also take in volumes of liquids and gases. Whenever you press on the plunger, you want to make sure to go nice and slow. You don't want to do it very fast. Otherwise, you'll create, like, for example, if you're trying to transfer a liquid solution, then you'll create bubbles if you try to press this plunger up and down too fast. And I'll show you what that would look like with a single channel pipette during the demonstration that I give. And then the last important note, at least the last important note that I can think of, is to make sure to dispose of pipette tips into biohazard sharps containers. And the reason for that is because Environmental Health and Safety at ASU deems pipette tips to be sharps. So we want to make sure to eject them into a sharps biohazard container immediately after we're done using the pipette tips. And next, I will be giving a demonstration. One of the main reasons that I decided to create a video for this presentation is so that I could be able to provide this demonstration while I'm in the lab, since I didn't really want to take any laboratory equipment outside of the lab space, since EHS, I believe, thinks that that's a no-no. So anyway, that'll be the next part of this video. In our setup here, we have a box of pipettes, which have a volume range of 2 to 200 microliters. Next, we have a box of 20 microliter pipette tips. Typically, if there is one number on a pipette tip box or on a pipette, that number is the maximum volume that it can transfer in units of microliters. We are working with DI water in this demonstration, and no matter what liquid solution you are or will be working with, it is important to have a paper towel or absorbent nearby in case of a spill. This pipette reads 200, meaning that the maximum volume that it can transfer is 200 microliters. To adjust the volume for an Eppendorf pipette, rotate the adjustment dial at the top of the pipette. Make sure that whatever vessels or containers that you are transferring fluids between are properly labeled. To start, open the pipette tip box, obtain the pipette tip by pressing the bottom of the pipette into the top of a pipette tip, and then close the box. Set the pipette down and open 2 containers. On pipettes, there are 3 different positions at which the plunger at the top can be: the rest position, the first position, and the second position. The rest position means that the plunger is not depressed at all. The first position is when the plunger is depressed but not all the way, and the second position is when the plunger is depressed all of the way. When you work with a pipette, you can figure out where the first position is because there will be some resistance at the first position. When trying to get a fluid into the pipette, press down to the first position, then depress the lower half of the pipette tip into the solution, then draw it out of the fluid by slowly lifting your thumb up until the plunger is back in the rest position. Make sure not to press down to the second position during this step, because if you do, you will draw in more liquid into the pipette tip than you intend, and therefore you would have an inaccurate volume measurement, and some of the liquid could end up going inside of the pipette and potentially damage the pipette. Then wipe the pipette tip on the side of the container to make sure that you do not have any excess liquid on the edge of the pipette tip. Then, to eject the fluid into a different container, depress the pipette tip into the container and press slowly down to the second position, meaning depress the plunger all the way, to completely remove the fluid from the pipette. Throughout the process, make sure that you are holding the pipette straight up and down rather than at an angle, besides when you are wiping the pipette tip against the side of the containers. Close the containers or seal them once you are done transferring fluids, and eject the pipette tip into the nearest biohazard sharps container.